Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Let's solve one question from IIT JAM Biotechnology examination. Choose the right combination of components required to set up a PCR from the following. We have to set up a PCR reaction and what are the requirements is asked in a question. So the options are DNA template, 2 primers, T antipedes and DNA ligase. Another is DNA template, 2 primers, antipedes and DNA ligase. The third option is template RNA, 2 primers, antipedes and DNA polymerase. And the last option says template DNA, 2 primers, D antipedes and DNA polymerase. So let's go through the steps of PCR reaction and find the answer for the question. Here PCR is a polymerase chain reaction. This is carried out in a thermal cyclers and the program can be set for the reaction to occur. So here what happens in a thermal cycler? First we have to take the template DNA. This is a template DNA having 2 strands 5' to 3' and complementary strand 3' to 5'. In the first step of the PCR, there is a denaturation of this double-stranded DNA. This denaturation occurs at 93 to 95 degrees Celsius. So we need to set the program at this temperature and thermal cycler works by its own. Secondly, we have to add the primers. So primers annealing is done. This is done at around 50 to 70 degrees Celsius, accordingly which DNA we are taking for our reaction. So this is a primer annealing. After this, DNA synthesis take place. The temperature for which is 70 to 75 degrees Celsius. Now for DNA synthesis, we need D antipedes and also DNA polymerase. Here the temperatures are very high and for this we need to use DNA polymerase which can sustain at these temperatures. So the DNA polymerase used in a PCR is usually isolated from Thermophilus bacteria. The name is a Taq polymerase. We commonly use TAQ, Taq polymerase, and this is isolated from the bacteria Thermus aquaticus. So using this Taq DNA polymerase, DNA synthesis is done. Now this completes one cycle of the PCR. Secondly, here we find the 4 strands after first cycle. And in this way, around 25 to 30 cycles are carried out to get enough amount of DNA for further verifications or further experiments what we need to perform. So again, these DNA will get denatured and whole cycle will get carried out in a thermal cycler and we can get enough amount of DNA. So when we focus on the requirements, what are the requirements? We need a template DNA, then we need primers, then there is a requirement of DNA polymerase and D antipedes are also required for the formation of DNA. All these requirements are added in a buffer and it is kept in a thermal cycler which gives us polymerized DNA or we can say more number of DNA strands after the completion of cycles. So with this discussion we find the answer for the question is option D. For PCR reaction we will require template DNA, 2 primers, D antipedes and DNA polymerase. Option D. So here I have concluded with today's video. I request you to please like, share and subscribe for my channel and also to hit the bell icon. Thanks for watching.